Welcome. My name is Matt Biggs. I'm a computational biologist at AgBiome. We're a crop protection company based in North Carolina, and one of our primary resources for discovering new solutions to agricultural pest problems is our large microbial collections. We have over 80,000 isolated bacteria, and for each one of those things frozen down in a freezer, we also have a corresponding genome. And we use that genomic information to help us gain insight into the microbes we discover and also to guide us, to help us make our assays more efficient and help us discover things more rapidly. For this project, we partnered with the Bill and Melinda Gates Foundation to identify solutions to these two problematic diseases, sorghum anthracnose and black sigatoka on bananas. Both bananas and sorghum are important crops worldwide and play a particularly central role in the diets of smallholder farmers in sub-Saharan Africa. Both diseases can cause yield losses of 50 to 100%, and there aren't a lot of options for controlling them. So we started this project by screening over 1,200 bacterial isolates against these pathogens on a detached leaf assay, and from that screen, we identified over 150 isolates with activity against one or both of the pathogens. You'll notice here, this is a picture of our workflow, and the idea with the detached leaf is it's an actual piece of leaf from the plant that's sprayed with both fungal spores and then treated with some of the bacterial culture. And from there, we can measure the progression of disease and identify those isolates that control the disease best. So this depiction here is of the cladogram of the relatedness between the microbes that were reproducibly active in this assay. And the first thing that was surprising for us was the great diversity of active microbes. So we discovered robust active microbes from 5 different phyla. And to put that in perspective, humans are in a different phylum from jellyfish, right? So this is very diverse set of microbes, all of them with activity. Some of those were not particularly surprising. So bacilli are some of these. This is the firmicutes in green, and over here we've got some of the pseudomonads. So those are well known to be fungicidal, and some of those strains are related to products on the market already. So not surprising, but a nice validation of our discovery approach. And then there are also some microbes and some taxonomic groups represented that were surprising, that for which we could find no literature evidence that they'd ever been reported as being fungicidal. So for example, we discovered some novel piney bacillus and some glutamacibacters, and that's all just interesting stuff to follow up on as we learn more about them. We continued screening, and we then implemented a machine learning approach. So both to gain insight into what was causing activity and also to help us discover new microbes. So we started with the genomes for everything that had gone through the screen, and we annotated the biosynthetic gene clusters, or BGCs, using anti-smash, and clustered those into closely related families using Bigscape. So that allowed us to shape our data set into something like this, where we had the strains, and for each one, the presence or absence of every BGC family, as we defined it earlier. And from there, we could associate presence or absence of a BGC family with activity against one or the other pathogens. So this table here is a subset of the strains of the BGC families that were associated with activity against sorghum anthracnose. And we did the association using a mixture of an enrichment method and a permutation feature importance method. Again, some of these were not surprising. So this BGC family here had homology to a well-known, well-characterized BGC that produces a molecule called fungicin. And again, down here, this one was homologous to a molecule called pyrrolnitrin. And both of those are well-characterized, well-known fungicidal molecules. So again, that's a nice validation of our discovery approach and our approach for selecting features. And then the exciting guys are these that say none. So those don't have any significant homology to known BGCs, and represent an opportunity for digging in deeper and discovering new modes of action. We then did some machine learning modeling up here, an in silico approach to show that these features are more than just patterns, nice patterns, but will allow us to discover new fungicidal microbes. So this was on a 100-fold cross-validation with a 75-25 training split, and we were able to show that 4 different machine learning approaches, from an enrichment approach, a couple random forest modeling flavors, and a deep neural network, all of them performed better than a random model. And so that was encouraging, and also allowed us to move forward and do this in the lab. So we used a couple of the best performing modeling approaches, specifically the random forest with oversampling. And we selected from the 80,000 plus strain collection at AgBiome, 176 isolates to go back into screening. From those 176, we discovered 32 additional strains that were active in the assay in the lab, and that was a much better hit rate than we would have gotten by a random chance alone. And we discovered a new genus that wasn't represented in the training set. So this Bacillus I was new and interesting for us. So for those who are interested, this paper will be on bioRxiv soon. And a lot of these strains now are in field trials across the world. So in Africa, the United States, and in Central America. So hopefully we'll have a good product here soon. Thanks for listening. Feel free to reach out with questions or just to connect, and stay safe and healthy. Thanks. Bye.